Have you ever wondered how researchers are developing the methods in which they study the anatomy and physiology of the human brain? Recently, there has been a scientific paper published that may revolutionize the standard practice into how the brain is studied. Researchers at UCLA and UC Irvine have begun looking into creating mini brain-like organoids that will be useful within studying the mechanisms of human brain development and neurological disease. The standardization of the development of mini brain-like organoids is important for studying disorders such as schizophrenia and autism spectrum disorder, where the brains of these individuals have similar anatomy. Although their physiology differs, mini brain-like organoids may also help researchers look into environmental factors in neurodevelopment, such as chemicals or viruses, as well as the Zika viruses. The theory behind the synthesis of mini brain-like organoids is through something called telecephalic organoid differentiation, using human pluripotent stem cells. More specifically, researchers are utilizing early-stage stem cells to produce these brain organoids in vitro, hoping that they will be a reliable prototype to study models of disease and be a great help at looking at neural structures. The production of these organoids includes taking human skin or blood cells and reprogramming them to become pluripotent stem cells. Pluripotent meaning that these cells are able to differentiate into any cell type within the body. The scientists can then direct these HPSCs to different cells and produce most cell types found within the brain, to then be influenced into aggregating into 3D organoids. The only problem is that there is no set and reproducible standardized practice in the methods that these researchers take to create mini brain-like organoids. The methods currently reproduce different results, where the scientists are hoping to create reproducible methods within telecephalic organoid differentiation with the development of a common protocol. The development of telecephalic organoid differentiation included determining which stage of early HPSCs would reproduce the same results more than other HPSCs. The stages being naive, intermediate, and primed. The different conditions of these HPSCs maintained under different culture conditions can exhibit variance in transcriptional states throughout the 3 different stages of HPSC. The best structural outcome came from naive formative pluripotency than organoids derived from HPSCs from later stages. The researchers have determined that the best structural outcome of the brain organoids came from the early developmental stage of the stem cell as opposed to the later stages, where they were more likely to produce the same results. It was also found that there are specific genes that served as predictive indicators for the quality of the organoids that would be derived from the native pluripotency HPSCs. These genes are TFAP2C and KLF5. Researchers also looked into growing these stem cells in a dish with mouse skin with fibroblast feeders. This provides the early developmental stem cells with essential chemical signals and structural support that would help them expand and preserve their immaturity over time. Although there are limitations to this, as mouse cells could make organoids less suitable for the development of cellular therapies to replace disease or damaged neural tissues within humans. Other labs are working on different methods such as RNA sequencing and computational analysis to find these genetic differences between stem cells that could produce differing qualities in the organoids. For example, another lab was looking into the 4 molecules that are responsible for keeping the stem cell in a less developed state without the need of mouse skin fibroblasts. These researchers were additionally able to produce high-quality, well-structured organoids through these findings. To recap, the implications of these findings are extremely relevant as it is emerging technology in neurological research and can aid researchers with a more focused view of the human brain development and neurological disease. HPSC-derived brain organoids offer many different opportunities where researchers will hopefully soon be able to develop a reliable and reproducible method.